Chapter 26.1, we are going to talk about some things um, that may make some people feel uncomfortable. But here's the thing: it's not my intention to make you feel uncomfortable. It's my intention to educate you. If I refer to someone, if I refer in this section to a female having a vagina or a male having a penis, I'm referring to their biological sex and not the gender that this person identifies with. Two completely different things. Uh, I just want to make sure that that is out in the open, as clear as possible, that nobody is intended to be offended by this. This is simply biological education. So far, all the things we've talked about have been absolutely required for survival, right? How you can't live without a brain or a heart. However, the reproductive system is not necessarily um, required for an individual's survival. Although it does produce several important hormones, um, this system actually almost hibernates until an individual hits puberty. This is a period of life in which the reproductive organs mature and eventually become functional. And this chapter is going to explore the male and female reproductive organs, and we're going to look at how the reproductive systems are regulated and how these sex hormones influence other body systems. We're not going to discuss pregnancy or the functions of the female reproductive tract in this section. We're going to talk about that in chapter 27. We're going to start by looking at the main parts of the reproductive system, the process of cell division, and the production of gametes or sex cells.
Some basic cell biology review. As you guys know, in all animals, there are those two types of cell division. There is mitosis and meiosis. Mitosis is the cell division that all somatic cells undergo. These are body cells, muscle cells, skin cells, bone tissue, all of these undergo mitosis. But if you guys remember, there are some of those that are amitotic, like neurons. They're unable to perform this division or divide once they have differentiated. Now, mitosis is going to produce daughter cells that are genetically identical, right? They produce clones. Meiosis, however, is a special type of cell division that only occurs in sex cells, cells that are determined to become gametes. And we're going to look at how mitosis compares to meiosis, but let's look at some basic principles and terms associated with the reproductive system first. I'm sure you guys are aware males and females' reproductive systems differ in many ways, but they do share a number of similarities. In both males and females, the most prominent and most important organs are the gonads, or the primary sex organs. In males, these are the testes, and in females, these are the ovaries. And these gonads secrete sex hormones. In males, most often, or most predominantly, it's testosterone, and in females, it's most predominantly estrogen. The gonads also produce gametes, or these sex cells, through the process of meiosis. In males, these gametes are sperm, and in females, these gametes are ova, often called egg cells. And then both males and females have additional organs and structures that contribute to their function, and these are known as accessory reproductive organs. And the similarities reflect between the male and female. These similarities reflect their similar function. So if you guys keep that in mind, that should help you as we go through this chapter. So each human somatic cell tends to have 46 chromosomes, paired so that there are 23 pairs of chromosomes in each nucleus. These are called diploid cells, meaning that they have two sets of chromosomes, or they're at 2n. Their number of chromosomes is considered, quote unquote, 2n. And that's because these chromosomes are paired. Now, one set, or half, 23 of these chromosomes, are going to consist of the paternal chromosomes that came from the father of the individual, and the other set is the maternal chromosomes that came from that individual's mother. And then each of these Each member of a pair of chromosomes tends to have the same genes, and these genes are going to be segments of the DNA that code for a specific protein. And so for this reason, the pairs are called homologous chromosomes, otherwise meaning sharing a similar structure. You can see some homologous chromosomes here, and you see the two sets of paternal in purple and the two sets of maternal, or the set of maternal in blue, light blue. However, the matching genes on these homologous chromosomes can have different variants. So the two genes for these two paternal genes, or these two homologous chromosomes you see here, can have different alleles. These are the variants of the genes. For example, let's say that let's say the paternal's gene here has the gene for hair color. The paternal chromosome might have the allele for brown hair, where the maternal chromosome might have blonde hair allele. And we'll talk about how that works in the next chapter, but the understanding of how this chromosome is shaped is important. Now, during fertilization, this process during which a sperm and an ovum fuse, during the process of which sperm and ovum fuse, there's going to be a single-celled offspring called a zygote. A zygote is a cell that will eventually divide to produce all of the cells in a new individual. And so for this reason, it has to contain the correct number of chromosomes. It has to have 46, or 2 pairs of 23. And this means that a sperm and an ovum each have to contribute 23 chromosomes. Otherwise, the zygote would end up with 92 chromosomes, and that's way too many. The human would not be adequately formed. And this is made possible by the process of meiosis. Meiosis divides these daughter cells to have half the number of chromosomes. They're haploid. So each sperm has 23 chromosomes and each egg has 23 chromosomes. Combined together, they have the 46 required, and they pair up as 23 pairs. Now, as with mitosis, just before meiosis begins, all of the DNA in the cell is replicated so that each new cell gets a complete copy. The result of DNA replication is a cell with 2 identical copies of its chromosomes, each called sister chromatids. Each pair of sister chromatids tends to be connected in the center by a centromere, which you can see here in the center of this X shape. This is a centromere holding together 2 sister chromatids. Now, if you guys remember from chapter 3, when you talked about a cell not dividing, its DNA is in the form of chromatin, right? Which is just one long piece of DNA with all of its associated proteins. After replication, when the cell division begins, that chromatin is going to densely coil, and it's going to turn itself into these bar-like chromosomes. And then also, like mitosis, you're going to see that meiosis has 4 phases: prophase, metaphase, anaphase, and telophase. But in meiosis, each of these occurs twice. So meiosis has 2 divisions. There's meiosis 1, otherwise known as the first meiotic division, and meiosis 2, or the second meiotic division. Now, this first meiotic division is going to separate the homologous pairs to produce what are called haploid cells, or 1N cells. These only have half the number of chromosomes of the original cell. And so for this reason, this process is sometimes called the reduction division. And at the beginning of prophase 1, The DNA of these chromosomes has already been replicated. Now the chromatin condenses so that individual sister chromatids now become visible through a microscope as those thin lines. At the same time, the nucleolus disappears, the nuclear membrane breaks down, and the spindle fibers and centrioles form and align on opposite ends. In middle to late prophase 1, you've got the homologous chromosomes moving together and pairing up at tightly, side by side in a process called synapsis. And when the two homologous chromosomes are touching, they are called tetrads. While touching, some sister chromatids may break in one or more places, and the homologous chromosomes exchange segments of their DNA, forming new chromatids with new combinations of genetic information. This is a step called crossing over, and it's actually vital for genetic variation. Now, this is the first process that shuffles the alleles. And so this changes the chromosomes so that their sister chromatids are no longer identical. And it results in these genetically unique cells at the end of meiosis. By the time we get into metaphase 1, the sister chromatids of the homologous pair line up on either side of the equator or the midline, forming a double line of chromosomes. Spindle fibers then branch out from the centrioles at opposite ends of the pole and attach to each set of sister chromatids at the centromere. Do note that the orientation of the paired chromosomes tends to be random with respect to which chromosome from the pair is on one side of the equator or the other. For example, any combination of maternal chromosomes may line up on one side with the remainder on the other, and then the same random combination occurs with paternal. So you can get a completely random set of maternal and paternal chromosomes in this first division. Now in anaphase 1, you're going to see one set of sister chromatids from each pair separate from its partner as they are pulled to opposite poles of the cell from the spindle fibers, okay? And then due to the random orientation during metaphase 1, separation is random as well. This process is called independent assortment. And this independent assortment is the second shuffling of alleles, further increasing genetic variability of the daughter cells. Now that the sets of sister chromatids have arrived on opposite ends of the cell, it's time for telophase 1. And this is when a nuclear membrane may reform, but this doesn't always happen. Regardless, cytokinesis is eventually going to take place, dividing the cytoplasm, separating these two new daughter cells. These are two genetically different haploid cells with 23 individual homologous chromosomes, each of them having a pair of sister chromatids. So this is these two cells are genetically different due to that random crossing over and the independent assortment having taken place. And here you can see I put all these individually. I put them all together so that you can see the flow of these chromosome assortment. Now there can be some confusion as to why the daughter cells at the end of meiosis 1 are haploid, because they appear to have the same number of chromosomes as the starting mother cell, right? Now, remember, you're not seeing 4 individual chromosomes, you're seeing 2 sets of sister chromatids. And a set of sister chromatids is simply 2 identical copies of one homologous chromosome. A simple analogy here, if you think of our class, you have 2 textbooks, right? The class textbook and the lab manual, with each one having similar information, but it's slight variation. So the 2 books represent a pair of homologous chromosomes found in a normal diploid cell. each one containing similar but slightly different information in its DNA. Now imagine that the JSU bookstore sends you 2 copies of the lab manual instead of a single separate lab manual and a class textbook. You now have 2 identical copies of the lab manual, a single book, not 2 individual books. And this represents a pair of sister chromatids. They are simply 2 copies of the exact same genetic information. We have a figure here that shows you this example. So you can see these 2 homologous chromosomes in a diploid cell. They're going to undergo DNA replication, and then you've got 2 copies of these homologous chromosomes, right? Now, this is still a diploid cell because the number of chromosomes technically has not changed. Eventually, we get to meiosis 1, and you see the 2 copies here of the 1 homologous chromosome flows into this first daughter cell, and then the other copy flows into the second daughter cell. So the diploid mother cell has those 2 homologous chromosomes. It has the lab manual and the class textbook. But by the end of meiosis 1, the resulting cells have only 1 replicated homologous chromosome. They have 2 copies of each book, right? Either the lab manual or the textbook. And so the number of distinct chromosomes has been reduced by half, from 2 to 1, making the cell now haploid. We've really simplified this because you can see that the sister chromatids are not necessarily identical because of the crossing over and individual assortment that occurs. The basic principle remains the same. A set of sister chromatids represents 1 homologous chromosome. Now, the second meiotic division, or meiosis 2, is going to separate the chromosomes of each. It's going to separate the chromatids of each chromosome. And the resulting cells will still be haploid, but they're only going to have 1 copy of each chromatid rather than 2 copies. And so the same way, we have the 4 stages: prophase 2, metaphase 2, anaphase 2, telophase 2. Prophase 2 is the second prophase event is going to be the same as the first prophase event. It occurs in both cells produced by meiosis 1. The nuclear membrane, if it did reform, breaks down. The sister chromatids remain condensed. During metaphase 2, each pair of these chromatids aligns to form a single line across the equator of the cell. The spindle fibers extend from the centrioles to each sister chromatid, and they attach at the centromere. Anaphase 2, the sister chromatids from each chromosome now separate during anaphase 2 and are pulled to opposite ends of the cell. Each sister chromatid is now a chromosome or a non-replicated chromosome. Cytokinesis will generally begin during anaphase 2. And then during telophase 2, these non-replicated chromosomes arrive at opposite ends, the nuclear membrane reforms, cytokinesis continues, and the spindle fibers and centrioles disassemble. What started with 1 original cell has now become 4 genetically unique daughter cells, each with 23 non-replicated chromosomes. In males, these 4 daughter cells mature into 4 sperm cells. In females, only 1 daughter cell goes on to become an ovum. But the basic steps of meiosis are the same for sperm and ovum production, as they're necessary to achieve the correct number of chromosomes and ensure genetic variability. The specifics of sperm and ovum production will be discussed later. And here again, you can see this figure shows the process through meiosis 2, how we go from 2 sets of haploids, 2 sets of sister chromatids, and we break those down to become 4 genetically unique haploid daughter cells. Now, at first glance, mitosis and meiosis look alike, so let's compare them side by side. First thing to consider is, why do they occur? Mitosis creates new somatic cells for either tissue growth or repair, and so these new cells have to be genetically identical to the original diploid cell. In contrast, meiosis produces gametes, right? Sperm and ova for reproduction. These cells have to be haploid, and they have to be different combinations of the genes in order to produce a genetically unique gamete. Now we know the why, right? So we can apply it to how the processes occur. So mitosis is going to begin by making a complete copy of the DNA, after which the cell is divided so that each cell gets one copy. The result is two genetically identical diploid daughter cells. Meiosis also begins with DNA replication, but it goes through meiosis 1 and meiosis 2. So it has those unique sets of steps that reduce the chromosome number and shuffle the alleles. Meiosis 2 then completes the process to form 4 genetically unique haploid daughter cells. So if we look at this figure, on the left side, you see mitosis. On the right side, you see meiosis. So mitosis, you have the mother cells duplicating the DNA, and then after metaphase, you see them all lining on the plate, and each of the daughter cells that's produced is a diploid daughter cell, genetically identical to the mother cell. On the right side, you see a mother cell duplicating its DNA, and then through prophase 1, you see the crossing over. So these tetrads will give a piece of their DNA to a homologous chromosome next to it, and so now they're genetically unique. And then in metaphase 1, they are aligned randomly. It could be both paternal chromosomes. In this example, you see the paternal and the maternal designated by blue and purple. It could be both blues line up on the same side, and so those separate out. It could be a blue and a purple. That's the genetic variability that's introduced here in this individual assortment. And then you get to prophase 2, where this meiosis 2 sort of works similarly to mitosis, insofar that it separates these into individual chromosomes. But remember, we're looking at a pair of remember, you're not looking at a pair here. You're looking at you're looking at 2 copies of one chromosome. So you see, this is not a pair of chromosomes, rather a set of copies of one chromosome. And then they differentiate, and again, you have that genetic variability created from crossing over and individual assortment, resulting in 4 individually unique daughter cells. But for now, that's enough about cell biology. Let's talk about anatomy, the anatomy of the male reproductive structures. And so the first thing we're going to look at are the paired testes, or the male gonads. These testes produce sperm, which travels through a series of passages known collectively as the duct system, including ducts which go through the penis, and that's the next topic we discuss. Then we'll move on to the accessory glands, which secrete fluids into these ducts, and that becomes part of semen, this fluid that accompanies the sperm. Finally, we conclude that the external structures, including the scrotum and the spermatic cord. So we're going to go through all of these in this unit. This is a solid overview of what we're going to discuss, all the way from testes to the eventual penis and the urethral opening. So that path that the sperm cell travels will be crucial to understand, as well as all the organs that facilitate its travel. Now, these paired testes, a singular one is called a testis. Colloquially, they're called testicles. Uh, these are ovoid structures that could be about 4 centimeters long and is 2.5 centimeters wide. Now, they're located outside the abdominopelvic cavity in a sac-like structure called the scrotum. And the scrotum is going to be composed of skin, connective tissue, and smooth muscle. And this connective tissue surrounds each testis and divides it internally into about 250 internal segments known as lobules. Each lobule contains 1 to 4 tightly coiled, looped seminiferous tubule. And these tiny tubules are going to be the sites of sperm production. The testes perform 2 vital functions. They produce sperm cells and they secrete androgen hormones, most importantly, testosterone. The seminiferous tubules contain 2 types of cells, including the spermatogenic cells and the sustentacular cells. Now, the spermatogenic cells form sperm, and the sustentacular cells have several functions that support the production of sperm. One of them is the production of testicular fluid, which contains nutrients for these developing sperm cells. In between these seminiferous tubules, you're going to find interstitial cells, which produce and secrete androgens, mostly testosterone, into the surrounding interstitial fluid, hence their name. Interstitial cells are in the interstitial space, secreting into the interstitial fluid. Additionally, these seminiferous tubules are going to be surrounded by muscle-like myoid cells, and these contract and push sperm and testicular fluid through the tubules and out of the testes. The seminiferous tubules of each lobule merge into a single straight tubule that moves this sperm into the rete testis. And this rete testis is the network of tubules on the posterior side of the testis. Sperm is going to pass through efferent ductules and then they're going to enter this first portion of the duct system, the epididymis. Now, each testis is going to receive blood from a testicular artery that branches from the abdominal aorta superior to the pelvis. You're going to find a network of veins called the pampiniform venous plexus. This pampiniform venous plexus drains the blood from the testes into testicular veins, and these testes are innervated by both sympathetic and parasympathetic divisions of the ANS. In addition, you're going to see a high number of nociceptors, pain receptors, and thermoreceptors, and these are concentrated in the testes and the surrounding connective tissues. These nociceptors explain, this high concentration of nociceptors tends to explain why testicular injuries are often so painful. So here's an image of the things we were speaking of. You see the seminiferous tubules and the sustentacular cells and the spermatogenic cells. You see interstitial cells lying in between them. Around them, you have the myoid cells. Eventually, you see all of these being pushed into the straight tubule, and the straight tubules drain into the rete testis. If you guys remember the rete testis, this network of tubules on the posterior side of the testis, and then sperm will pass through the efferent ductules into that epididymis. And you see here some of the blood flow as well. You see a testicular artery. You see some of the pampiniform venous plexus draining into a testicular vein. and there is innervation, but the nerves are not shown in this image. A couple of test yourself questions. Complete this analogy. Testes are to gonads as sperm are to blank. So testes are gonads. What are sperm? Sperm are gametes. Gametes are those sex cells, sperm and ova. Now, what process shuffles the alleles and causes homologous chromosomes to no longer be identical? This process shuffles alleles and causes homologous chromosomes to no longer be identical. It's crossing over. Remember, the crossing over exchanges those bits of DNA and makes homologous chromosomes no longer identical. Now, once sperm actually leaves a testis, it travels through a series of ducts. It's going to travel through the epididymis, the ductus deferens, the ejaculatory duct, and eventually the urethra. And this duct system transports the sperm and is involved in ejaculation, the process through which semen is expelled from the penis. As sperm moves through these ducts, they are supported and nourished by secretions collectively known as semen from the accessory structures. But first, we're going to talk about the epididymis, because the epididymis is the site of sperm maturation and storage and is filled with many, many ductules. It consists of a single tube called the ductus epididymis that passes through all the regions of the epididymis. And the ductus epididymis is pretty long. It's about 6 meters long, but it is so tightly coiled and twisted that it manages to fit inside a less than 4 centimeter length of the normal epididymis. Each epididymis sits on the superior posterior surface of the testis and includes a head, body, and tail region. The head of the epididymis sits on the superior surface containing the efferent ductules of the testis, and sperm moves from the head to the body of the epididymis, which is its narrow mid portion, and then eventually into the tail, which is its smaller inferior portion. And the mucosa lining this hollow lumen of the ductus epididymis, it tends to be lined with pseudo-stratified epithelial cells with long, non-motile microvilli, or stereocilia. These absorb excess testicular fluid, and then they pass nutrients to the sperm in the lumen. This helps to complete the maturation of the process of the sperm. And the long length of this ductus epididymis allows ample time for the sperm to mature. It also allows sperm to be stored for up to several months if ejaculation does not occur. We're going to look here in a little bit, and you're going to see that sperm that are not ejaculated in those months tend to be reabsorbed. Now, the ductus epididymis will widen within the tail of the epididymis and become the ductus deferens, often called the vas deferens. The ductus deferens is pretty long, between 40 and 50 centimeters, and very thin, about 2.5 millimeters. Now, it's going to extend along the posterior border of the epididymis inside the spermatic cord with the testicular arteries, veins, and nerves. This ductus deferens enters the pelvic cavity through a mostly fibrous tunnel called the inguinal canal, after which it passes along the lateral side of the bladder over the ureter and then along the posterior bladder. Here is it's where it's going to end in a sac-like wider area called the ampulla. Now I got a couple images here I wanted to show you guys, make sure you understand. So this region here is the head of the epididymis. Then you have this middle narrower body of the epididymis, all the way down to the tail of the epididymis. And eventually the tail is going to end up in the ductus deferens you see here. And you just see this follows this path to become the ductus deferens. And it's just the same tubule. It just has a different name now. And yes, this does pass through around the bladder. As you can see here, this epididymis joins up with the ductus deferens, and the ductus deferens here runs along the lateral side of the bladder up onto the ampulla, and you can see this is the ampulla of the ductus deferens here. So we're going to refer back to this image periodically just to make sure everybody's following along. Now, the organization of tissue for the ductus deferens is pretty much the same as you've seen for other hollow organs. There's an inner mucosa, a middle layer of smooth muscle, and an outer adventitia. The mucosa of the ductus deferens consists of pseudostratified columnar epithelial cells resting on a loose connective tissue basilar layer. And the muscularis here is going to consist of 3 layers of smooth muscle. The inner and outer are longitudinal, and the middle is circular. During ejaculation, this smooth muscle creates strong contractions that rapidly squeeze sperm forward along the tract. Like the epididymis, the ductus deferens can store sperm for several months and reabsorb any that is not ejaculated in that time. After they pass through the ampulla, the sperm next travel into the short ejaculatory duct, which is about 2 centimeters long. This is located at the junction of the ampulla of the ductus deferens and the duct of an accessory gland called the seminal vesicle. Each ejaculatory duct is then going to travel through another accessory gland, the prostate gland, and it empties into the urethra. And then the urethra, if you guys remember, is the length of the penis, essentially, where we're going to connect from, normally from the urinary bladder to the external body surface. In males, this does move both urine and semen, but not at the same time. So it belongs to both the urinary and the reproductive system. It does have 3 regions. There's the prostatic urethra, surrounded by the prostate gland, the membranous urethra, passing through the external urethral sphincter, and the spongy urethra, extending through the penis and terminating at the external urethral orifice. Now, as with the ductus deferens, the smooth muscle in the wall of the urethra contracts during ejaculation to help expel this semen through the external urethral orifice. The mucosa of the urethra contains several small mucus-secreting urethral glands that empty into the urethra and help contribute to the composition of the semen. Now, the penis itself delivers sperm into the female reproductive tract and is therefore called the male copulatory organ. At the base of the penis, this is called the root. The body or the shaft enlarges into the end of the penis called the glans penis, under which you're going to see is where the external urethral orifice is located. There is skin that covers the penis, and it tends to be loose and forms a circular fold called the prepuce, or foreskin. And this is around the glands of the penis. The prepuce can be removed shortly after birth in a procedure called circumcision. And there is a great deal of controversy about the medical necessity of circumcision. Many studies show that it serves no measurable benefit. Now, internally, the penis contains 3 cylindrical erectile bodies, or corpora. Individually, these are called corpus. Now, these are sheaths of dense fibrous connective tissue, which you can see in a transverse section that I'll show you in a minute. But each erectile body is essentially a spongy network of connective tissue and smooth muscle filled with vascular spaces. And the first two erectile bodies are the paired dorsal corpora cavernosa. And at the base of the penis, the two corpora cavernosa split to form the two crura of the penis that attach it to the ischial rami. Now, the other erectile body is the ventral corpus spongiosum. And the corpus spongiosum expands to form the bulb of the penis at the base. In a moment, you guys are going to see how the bulb sits between the two crura, and together these structures make up the root of the penis and connect it to the pelvic bones. The corpus spongiosum also expands at its distal end to form the glans. And during sexual excitement, the vascular spaces fill with blood, causing the penis to enlarge and become rigid. This is called erection, as we'll talk about in a minute. Now, the blood supply comes from the internal iliac arteries, which give rise to internal penile arteries, and these internal penile arteries divide and form dorsal arteries and deep arteries. The dorsal arteries supply the penile skin, fascia, and corpus spongiosum. The deep arteries then feed the corpora cavernosa. And the penis is innervated by many sensory and motor fibers, both sympathetic and parasympathetic. There's tactile, pressure, and temperature sensors in loads here. These are very important for sexual stimulation, as well as control of muscles that regulate the distance from the scrotum from the body wall. And you can see here a cross-section of the penis with the dorsal section on top. You can see the two corpora cavernosa, the paired corpora cavernosa here, with a corpus spongiosum down here on the bottom. And you can see that the urethra, the spongy urethra, travels inside of the corpus spongiosum. And you do see that the deep arteries feed the corpora cavernosa, and the dorsal arteries feed the external layers and the corpus spongiosum. Now, together, the penis and the scrotum make up the external male genitalia. They are part of the male perineum, and this perineum is a diamond-shaped area between the thighs, bordered by the pubic symphysis and the ischial tuberosities and the coccyx. Okay, and this perineum is separated into the urogenital triangle, consisting of the base of the penis, the scrotum, and then there's the anal triangle, which consists of the anus. And the floor of the perineum is formed by muscles, which you guys should have already covered in the muscle chapter. So if we do look at this image, you notice first we're going to look at the root of the penis, and you see it has the crus and the bulb, and you can see that the bulb is here, and the crus, the paired crura, are here. Each of those being made out of the root. You also have the body of the penis, which is just this section here, essentially, and then the glans, or the head of the penis, typically covered in that foreskin or prepuce. And these, again, these can be removed, but that's essentially a cosmetic surgery at this point. But I just wanted to, again, we're referring back to this image to make sure everybody understands what structures we're talking about. Now, the accessory sex glands are essentially exocrine glands for the male reproductive system. Since they produce most of the liquid portion of semen, this mixture of secretions and sperm that is expelled during ejaculation, and they help lubricate the outside of the penis during intercourse. The accessory glands include the seminal vesicles, the prostate, and the bulbourethral glands. Now, The paired seminal vesicles are going to be found on the posterior surface of the urinary bladder near the ampullae of the ductus deferens. Each vesicle is about 15 centimeters long, but is tightly coiled to be about a third of the length. The duct of each seminal vesicle will converge with the ductus deferens to form that ejaculatory duct. And the exterior of each gland now has a fibrous capsule that covers a thick layer of smooth muscle. Now, deep to this mucosa layer, you're going to find, deep to this is the mucosa layer that you're going to find is made of a pseudostratified columnar epithelium working to secrete seminal fluid, which is a yellowish secretion making up about 60 to 70% of total semen volume. And this seminal fluid contains fructose, which is a sugar that the sperm uses as a nutrient. It contains prostaglandins. Prostaglandins stimulate smooth muscle contractions in both male and female reproductive tracts. This increases the sperm's viability. And it also has coagulating proteins and enzymes. A coagulating protein together with an enzyme from the prostate forms a temporary clot of semen in the female reproductive tract. Now, in addition, the pH of the seminal fluid tends to be more basic or alkaline, and this helps neutralize the female vaginal fluid's natural acidity. Then you have the prostate, and this, be careful not to call it the prostrate, because that's not the same thing. The prostate is an egg-sized gland sitting just under the urinary bladder, and this surrounds the urethra and the ejaculatory ducts. Now, it is covered by an outer fibrous capsule and made up of about 20 to 30 tubular glands embedded in smooth muscle and dense connective tissue. During ejaculation, prostatic smooth muscle contracts and squeezes the prostatic secretions into the prostatic urethra through several ducts. The prostate can enlarge with age, which you guys will learn about when we talk about benign prostatic hyperplasia and prostate cancer. But normally, the prostatic secretions tend to be sort of milky, and they make about 20 to 30% of the semen volume. They contain citrate, which is another sugar that the sperm use. It also secretes prostate-specific antigen, PSA, and other enzymes which work to dissolve the clot of semen that initially forms so that the sperm can move deeper into the female reproductive tract. And it also contains antimicrobial chemicals which can inhibit the growth of some bacteria and decrease the risk of infection in the female reproductive system. Now, pH of the prostatic fluid is also more alkaline, and again, this works to neutralize acids from the male urethra and the female reproductive tract. All of these working together to give sperm a better fighting chance to make it to the egg. Now, the bulbourethral glands, or the Cowper glands, are these small marble-sized glands at the base of the penis on either side of the membranous urethra. And what they do is essentially they secrete a thick alkaline mucus-like fluid that works to neutralize any acidic urine that remains in the urethra before ejaculation. And it also lubricates the urethra for the passage of ejaculate, and it lubricates the glands of the penis during intercourse. And unlike the prostate gland, bulbourethral glands tend to grow smaller with age and are very small in older men. This is a sort of pre-ejaculate to sort of ready the stage for the sperm and the seminal fluid that will be flowing through here eventually. And so if we start here, we can see this short bit here is the ejaculatory duct right up until we hit the prostate, at which point it becomes the prostatic urethra. Again, you can see the prostate here, And down here, as we move toward the distal end of the penis, you have the bulbourethral glands secreting that pre-ejaculatory fluid, lubricating the urethra and neutralizing any acid. You also have the seminal vesicles here, again secreting fructose, and then the prostate. The prostate secretes its citrate. All of these fluids converge and are ejaculated as semen, in combination with the sperm cells, which have traveled up the epididymis, or from the epididymis up the ductus deferens and joined up after the ampullae of the ductus deferens into the ejaculatory duct. Now, despite the terms being somewhat ubiquitous, semen does not equal sperm. Semen is the milky white, sticky mixture of sperm and fluids from the testes, seminal vesicles, prostate, and bulbourethral glands. Now, sperm only contributes about 5% of the total volume of semen, and each typical ejaculate, which is the amount of semen expelled during one ejaculation, can be between 2 and 5 mils in volume and contain between 40 and 750 million sperm cells. And it seems like this is a large number, but only a small fraction ever reach the ovum. 5 minutes after sperm is ejaculated, it coagulates because of that clotting protein from the seminal vesicles and the clotting enzyme from the prostate. This coagulation prevents the sperm from leaking back out of the female reproductive tract. Between 15 and 30 minutes later, the clot breaks down because of PSA and other anticoagulant enzymes, and as it dissolves, the sperm are then gradually released from the clot, activated, and begin their ascent up the female reproductive tract. And the alkalinity, this alkaline pH of the semen, helps make the sperm fully motile, and the sperm can begin the process of capacitation. And by this process, the sperm undergo changes that enable them to penetrate and fertilize an immature female gamete. They finish this capacitation in the female reproductive tract, where the secretions will prepare the sperm's plasma membrane to fuse with that of the ova. And prostaglandins from the prostate are thought to thin the mucus here. They thin the mucus in the female reproductive tract and may stimulate some contractions, which pull the semen further in, helping to facilitate this fertilization. There's also the antimicrobial chemicals from the prostate killing some bacteria, including E. coli bacteria, which live in the large intestine and could cause illness if allowed to colonize in the reproductive tract. Now, for a man, the prostate can slowly enlarge from birth to puberty and then rapidly expand until about 30 years of age. Further enlargement can occur after 45, but if the prostate is non-cancerous and it still expands to the point of pushing on the urethra, this condition is called benign prostatic hyperplasia, or BPH. And sometimes this enlarged prostate can be secondary to prostate cancer, which tends to be the second most common cancer in U.S. men after non-melanoma skin cancer. And prostatic cancer is one of the leading causes of cancer deaths among men of all races. Risk factors include genetic predisposition, African ethnic heritage, age over 50, obesity, and poor diet. And prostate cancer screenings usually include a digital rectal examination in which a medical professional palpates or touches the prostate through the anterior rectal wall, and an assessment of the blood prostate specific antigen level. So in combination, this PSA level assessment and the digital examination the medical professional tends to be able to determine if this prostate is enlarged and whether a cancer screening that is necessary. Now, a PSA level below 2.5 nanograms per milliliter tends to be normal. If for any reason the number of cells in the prostate increases in either BPH or prostatic cancer cases, the PSA levels rise. And if there is any, sort of suspicious area like a lump, or if the PSA level is over 4.0, tissue biopsy specimens from different sites in the prostate need to be taken to look for cancerous cells. And prostatic cancer can be treated with surgery, radiation therapy, chemotherapy, or hormone therapy. And in some men, these tumors grow very slowly and have a low risk of metastasis. In these cases, regular monitoring may be all that is required. However, some tumors can be quite aggressive and metastasize to other tissues very rapidly. And so for this reason, screening and early detection are critical for them. Prostatic cancer is a killer for men, and the taboo around the digital examination tends to be the fear of, I don't want to get screened because I don't want a finger in my butt. And it's a silly taboo to be done away with, hopefully in the near future. Now, the scrotum is the sac that contains the testes, and it is located between the thighs. And you're going to find a midline septum that divides the scrotum into two internal compartments, one for each testis. And the location of the septum is marked externally by a ridge-like seam at the midline called the raphe. And this continues anteriorly along the ventral side of the penis and posteriorly to the anus. The wall of the scrotum includes an outer layer of skin and a layer of superficial fascia and a layer of smooth muscle called the dartos muscle. Here you can see the scrotum and this raphe, this midline, this seam that sort of runs down the midline, as well as the layers of skin, this outer layer of skin, this middle layer of superficial fascia, and an internal layer of smooth muscle called the dartos muscle. You can see here. And again, this is the only time I'm going to show this image, but I am going to refer back to it when we talk about spermatic cord and several other structures like the inguinal canal and things along that line. So just keep this image in your mind when we talk about things like the inguinal canal. And you can see that here, this external inguinal canal, just a sort of hole through the body wall there. Extending up from the scrotum is the spermatic cord, and this is essentially a tube made up of layers of fascia, and it contains the ductus deferens, which passes into the pelvic cavity. So the spermatic cord also contains blood vessels, nerves, and lymphatic vessels, as well as smooth muscle that's collectively known as the cremaster muscle. And the cremaster muscle controls the height of the testes. This all enters the pelvic cavity by passing through the external inguinal ring and ends at the inguinal canal. This is a passageway into the abdomen. Do note that sometimes the abdominal contents can protrude through the inguinal canal, and this is a condition called an inguinal hernia, and a condition about 25 times more likely to happen in men than in women, because of the large size of the inguinal canal and the external ring in males, necessary to accommodate the spermatic cord. Now, the scrotum and the spermatic cord together help support sperm production through regulation of the temperature, Now, the core body temperature of 37°C is too warm for producing large numbers of viable sperm. So under normal circumstances, the scrotum tends to be 3°C lower at 34 degrees C. And so this is the optimal temperature, and it is controlled in two ways. First, remember that the spermatic cord contains the testicular artery and an extensive network of veins, right? The pampiniform venous plexus, which surrounds the testicular artery before draining into the testicular veins. This unique anatomical arrangement of blood vessels results in a countercurrent heat exchange where the warm arterial blood passes into the testis and the cooler blood from the pampiniform plexus passing closer by, moving in the opposite direction, pulls some of that heat of the arterial blood away, making it a little cooler as it enters the testis. Secondly, the scrotum can be cooled or heated slightly by controlling its proximity to the body cavity. For when the external temperature is cold, the testes are pulled closer to the pelvic floor and the warmth of the body wall, and the scrotum becomes shorter and more heavily wrinkled. This decreases the surface area of the scrotum and reduces heat loss. But if the external temperature is hot, the scrotal sac relaxes and becomes flaccid or limp, and this increases the surface area for cooling, which may involve sweating, and it moves the testes further away from the warm body trunk. And these changes in scrotal surface area are controlled by the dartos muscle, which contracts to wrinkle the scrotal skin, and the cremaster muscle, which contracts to elevate the testes. In addition, the scrotum is innervated by many sensory and motor fibers, both parasympathetic and sympathetic. And in addition to the thermoreceptors listed previously, the scrotum has a high number of tactile receptors important in sexual stimulation. Now, the cremaster muscle tends to often contract when a man is sexually aroused in order to protect the area during sexual intercourse. This table you see here on the right shows each function of the individual male reproductive organs. And I've gone over all these, but I'll allow you to pause this and view it if you need to. We'll be moving on now. Now, sperm in the epididymis is going to travel immediately afterwards into the blank. Which one does it travel to after the epididymis? It travels into the ductus deferens, or the vas deferens. Remember that. So it's epididymis, ductus deferens. And then what does the contraction of the cremaster muscle do? Remember, the cremaster muscle does what? It pulls the testes closer to the body. If it's contracting, it's pulling them closer. If it's relaxing, it's going to allow them to float, to be flaccid and be pushed further away from the body. All right, now that we've looked at the anatomy of the male reproductive system, now let's turn our attention to its physiology. In specific, let's talk about how sperm cells develop. This is a process called spermatogenesis. And after that, we're going to look at the hypothalamus and the anterior pituitary gland and how they regulate the production of these hormones responsible for male reproduction. We're going to look at this male sexual response, the changes that take place in the males at puberty, and the change in males' reproductive functions that occur with age. And the first thing we need to look at is spermatogenesis, the process of male gamete development, sperm cell development. And this begins at puberty and continues throughout the entire adult's life. Every day, a healthy young adult male makes about 400 million sperm. And spermatogenesis is going to occur in the spermatiferous tubules of the testes through division of spermatogenic or sperm-forming cells. Spermatogonia are these stem cells that begin the process, and these are diploid cells with 46 chromosomes. They're going to be found along the basement membrane the basement membrane of the seminiferous tubules. And and before puberty, spermatogonia only divide by mitosis. But this is only in an effort to produce more spermatogonia. And then when puberty begins, they become functional stem cells and divide again, this time by mitosis, to maintain the stem cell line and create more spermatogonia. However, some of the cells resulting from mitosis differentiate into diploid primary spermatocytes. And as spermatogonia divide into these two cell types, the cell destined to become a primary spermatocyte is pushed toward the lumen of the seminiferous tubule while the spermatogonium takes its place along the basement membrane. Now, the primary spermatocyte is then going to undergo meiosis 1, forming two smaller joined haploid cells. These are called secondary spermatocytes. And then the two secondary spermatocytes rapidly undergo meiosis 2, producing 4 small haploid spermatids. Okay, and in this figure you're going to see, do notice that the spermatids are located at the lumen of the tubule. I'm sorry, I forgot to advance the slide. So yes, secondary spermatocytes are differentiated again and become the 4 spermatids. And you can see this figure here, and we can see all of these steps. What you see there labeled as 46 are just spermatogonia, and the spermatogonia there are undifferentiated there. And then eventually those will divide by mitosis, and one of those daughter cells becomes a primary spermatocyte. This primary spermatocyte is going to undergo the meiosis 1, producing those 2 haploid daughter cells, which again differentiate through meiosis 2 to become spermatids eventually. And those are at the lumen of the seminiferous tubule. So progress through this begins at spermatogonium, spermatocyte, and then spermatid, and the spermatids eventually become mature sperm cells. So spermatid is not quite the same thing. This is a spermatid, and this is a mature sperm. Now, in that figure you just saw, those spermatogonia were surrounded by this yellow substance in the image. Those are essentially sustentacular cells, or nurse cells, otherwise called Sertoli cells. These large cells extend from the basement membrane to the lumen of the tubule, and they're bound together by tight junctions. These tight junctions form a blood-testis barrier that separates the forming sperm cells from the immune system. Because if immune cells in the bloodstream encounter these new antigens on the genetically unique sperm cells, they're going to recognize them as foreign and mount an immune response. Notice, however, that the spermatogonia are outside the blood-testis barrier, and this is possible because they are genetically identical to other body cells, and so their antigens are not recognized as foreign, and indeed they have to be outside the blood-testis barrier for hormones and paracrines to reach them and stimulate that spermatogenesis. Now, in addition to forming the blood-testis barrier, sustentacular cells perform other functions, which explains why they're called nurse cells. These functions include providing structural support for spermatogonia development by maintaining the environment around the cells, secreting substances that stimulate mitosis of the spermatogonia, and initiating meiosis in response to testosterone and FSH production, providing nutrients to the dividing cells, phagocytizing damaged spermatogonic cells, and excess cytoplasm that may have been released from maturing spermatids, and then production of androgen binding protein, ABP, and inhibin, which help regulate spermatogenesis. Now you may be thinking, spermiogenesis, we just talked about that. And that's not quite the case. We talked about spermatogenesis. Spermiogenesis is the process of maturing the sperm, and it involves changing the size and shape of the cell. And spermiogenesis begins in the testes as the spermatid elongates and sheds any excess cytoplasm. As it continues, several other things happen. A structure called the acrosome, which contains digestive enzymes and helps fertilize the female gamete. This acrosome is gonna form over the nucleus, and more mitochondria are going to be produced and move to their positions around the beginning of the flagellum. The microtubules in the cell form the flagellum slash the tail. And these cells lose their connection to their sustentacular cells, and they're released into the lumen of the seminiferous tubule. All of this is spermiogenesis. The changing appearance of the cell as it matures from a spermatid to a sperm is going to be shown in a figure in just a second. But each sperm cell has a head, a midpiece, and a tail. And the head is what's going to include the nucleus and the acrosome with very little cytoplasm. The midpiece contains the mitochondria, and the tail is a flagellum that is surrounded by the plasma membrane. This tail moves in a mature sperm cell because of any ATP that is provided by the mitochondria in the midpiece. And we should see a figure of spermiogenesis here. So here you can see the development of a spermatid to a sperm. So after spermatogenesis, you have a spermatid, and it looks not that different than a regular cell. So this differentiation has to occur. These microtubules need to create a flagellum, and the mitochondria need to begin moving. And more of them need to be created. And also you can see this size of this cell begins to change, and the shape of it begins to change. In addition, you can see in this first one, you have this acrosomal vesicle. It begins to change shape as well as we move through. And the acrosome continues to just cover the nucleus as more and more of this change occurs. So eventually we get all of the mitochondria located in the midpiece. We have the acrosome fully covering the nucleus, and we have the flagellum fully formed. Well, now what we need to do is we need to get rid of all this excess cytoplasm, and so that's what it does, is it simply buds that off, and that will be reabsorbed by those nurse cells. The last step is sort of refinement, but the acrosome is fully formed in this image here on the right. You see the midpiece with its dense mitochondria, and the tail of the flagellum being fully realized. Now, even though the sperm has developed a flagellum, it doesn't actually activate it, but it will move to the epididymis and finish maturing. These sperm move very slowly, taking about 12 days to reach the tail of the epididymis, where they can remain viable for several months. But if the sperm are not ejaculated during this time, the body will resorb them and use those base products to create new ones. Together, spermatogenesis and spermiogenesis take about 60 to 70 days to complete. Now, one of the main hormones that regulates spermatogenesis and most of male reproductive physiology is testosterone. And testosterone secretion is going to be controlled by a multi-tiered negative feedback loop involving the hypothalamus, the anterior pituitary, and the testes. in a relationship called the HPG axis, the hypothalamic pituitary gonadal axis. And we've talked about this already when we talked about the neurology chapter. Now, we're going to look here at some events that produce and regulate testosterone and testicular function. First, we have the hypothalamus releasing gonadotropin releasing hormone. And this GnRH is going to stimulate the anterior pituitary to secrete gonadotropins. In this case, it's follicle stimulating hormone and luteinizing hormone, FSH and LH. In males, it is also called interstitial cell stimulating hormone, ICSH. Now, in addition to those, you have the testes, which are the target of the FSH and luteinizing hormone. The luteinizing hormone is going to stimulate the interstitial cells to produce testosterone, while follicle stimulating hormone stimulates the sustentacular cells to secrete ABP, androgen binding protein, and the hormone inhibin. Now, testosterone is the main hormone that stimulates spermatogenesis, but if you remember, testosterone-producing interstitial cells are located between the seminiferous tubules, but the target cells for spermatogenesis are spermatogonia and sustentacular cells, and these are located within the seminiferous tubules. So for this reason, sustentacular cells secrete androgen binding protein in response to follicle stimulating hormone, which in turn binds and concentrates testosterone in the seminiferous tubules around its target cells. Now, much of the testosterone also enters the blood, where it affects other tissues and leads to the development of male characteristics, which we'll talk about shortly. But when the level of testosterone rises to a certain level, gonadotropin releasing hormone secretion falls by negative feedback, which in turn causes a decline in FSH and LH levels. Now, in addition to these, the hormone inhibin secreted by the sustentacular cells decreases the secretion of FSH. Now, with this, sustentacular cells also convert some of this testosterone into estrogens, which directly inhibit testosterone production by the interstitial cells. And it can usually take a few years to establish this HPG axis, but once it is established, the testosterone level and spermatogenesis levels remain fairly stable until age 70. At 70, it's relatively common to see gradual decreases in both of these, both the testosterone level and the spermatogenesis level. Sometimes conditions can disrupt testicular function, leading to infertility, which we'll talk about in our clinical A&P section. So here you can see some of that hormonal regulation of the HPG axis. You see the GnRH release from the hypothalamus, which stimulates the anterior pituitary to release FSH and LH, which then go to stimulate testosterone production from interstitial cells and ABP production from sustentacular cells. Eventually, that ABP is going to bind testosterone, and the testosterone is going to stimulate more spermatogenesis and the development of those male characteristics. Now, male infertility is infertility rather, is the inability to produce a pregnancy after 1 year of unprotected intercourse. And approximately 40% of all infertility cases tend to result from male infertility, usually due to a low sperm count. Remember, normal counts are between 40 and 750 million sperm per ejaculate. Anything less than 15 million sperm cells per milliliter is typically indicative of infertility. And a low sperm count can result from any number of causes. Damage to the testes, like physical trauma, there's exposure to radiation, or maybe some underlying disease. And it could also be a developmental defect. During normal development, the testes begin forming inside the abdominopelvic cavity and then descend. However, sometimes this testis does not descend into the scrotum. This is called cryptorchidism. And the sperm cells in this case will not be produced. In addition to that, there's going to be inadequate secretion of gonadotropin releasing hormone, follicle stimulating hormone, luteinizing hormone, and testosterone. And any of those can also lead to lower sperm counts. So for fertilization to occur, sperm has to reach the oocyte, and this is deep within the female reproductive tract. Now, to optimize chances that fertilization occurs, sperm should enter into the female reproductive tract as far as possible. Typically, this occurs by sexual intercourse, often called copulation or coitus, and it is most commonly defined as the insertion of the male penis into the copulatory organ of the female. In our case, the human case, it's the vagina. And the basic phases of male sexual response include erection and ejaculation. Erection is going to be the enlargement and stiffening of the penis, which allows the penis to insert into and enter the vagina. An erection results when the erectile tissue in the penis becomes engorged with that blood. And then normally these blood vessels are going to be constricted, and so the penis is in a flaccid or relaxed state. But during sexual arousal, a parasympathetic reflex triggers the release of nitric oxide from blood vessels. And this nitric oxide will relax smooth muscle in the walls of the arterioles of the penis, dilating them. And so within the blood sinuses of the erectile tissue, this allows large amounts of blood to enter the tissue. In combination of increased blood flow and widening blood sinuses, this results in an erection. And the erection is maintained, at least partially, by compression of the veins that drain the penis. So there's a widening and a greater flow of blood into the penis, but the penis essentially clogs the drain and has to trap the blood there. Now, note that the corpus spongiosum does not become as rigid as the corpora cavernosa, and this allows the urethra to remain open during ejaculation so that sperm and semen can pass through. As I just said, yes. Now, erection does occur because of a variety of sexual stimuli like touch, mechanical stimulation of the penis, erotic sights, sounds, or smells. Emotions and thoughts controlled by the higher thought centers of the brain can also cause or prevent erections. But there is impotence or erectile dysfunction, and this is the inability to maintain an erection long enough to participate in sexual intercourse. We'll talk about that in the clinical A&P section for erectile dysfunction. But first, let's talk about ejaculation. This is the process by which semen is expelled from the penis. And although erection is under parasympathetic control, ejaculation is under sympathetic control. And it occurs in two stages, emission and expulsion. So in the emission stage, we move the sperm and testicular fluid, as well as secretions from the accessory glands, into the urethra. As semen accumulates in the urethra with the increase in sexual tension, the sensory impulses are stimulated and they pass into the sacral portion of the spinal cord. Motor neurons from the spinal cord stimulate skeletal muscles at the base of the erectile columns to rhythmically contract, resulting in the next stage, expulsion of the semen from the urethra. Orgasm tends to refer to the amount of time or this time period during which feelings of pleasure are experienced. In males, this orgasm coincides with ejaculation. Now, orgasm and ejaculation are quickly followed by resolution or relaxation, in which blood vessels in the erectile tissues constrict, forcing blood out of the penis, which becomes flaccid again. After ejaculation, a latent period or a refractory period follows. During this time, ranging from minutes to hours, a man is unable to achieve another orgasm. And as men get older, this refractory period tends to get longer. And there are a number of issues that can cause erectile dysfunction. Various psychological or physical factors like stress, depression, anxiety, or cardiovascular disease or diabetes mellitus. Obesity and tobacco use can also play a role, as well as certain prescription drug use. Older men tend to have a greater risk of ED because they are likely to have physical conditions also associated with it. The amount of connective tissue in the penis will increase with age, which reduces blood flow. And treatment for ED includes medications that permit blood vessels supplying the penis to remain dilated, thereby maintaining an erection. There are other treatments that may involve surgery to insert a penile implant or to repair damaged blood vessels. Now, and one of these medications that's actually commonly used, I'm sure you know Viagra. Viagra was actually a failed blood pressure medicine. They noticed that its primary effect instead was to engorge the those arteries in the penis while still constricting the veins, and so that Pfizer managed to pivot and say, okay, this doesn't work as well for blood pressure, but it does work really well for maintaining an erection. So let's sell that. Now, testosterone is known as the male steroid hormone for more reasons than stimulating spermatogenesis. Without testosterone, many characteristics we define as masculine or male would not be distinguishable. And this level of hormone increase at puberty will increase dramatically and is responsible for the growth, maturation, and maintenance of male reproductive organs, including the testes, scrotum, penis, and duct system. Now, testosterone also triggers the development of secondary male sex characteristics featured in the non-reproductive organs associated with adult males. And throughout a boy's childhood, testosterone from the testes and androgens is low, but sufficient enough to inhibit gonadotropin-releasing hormone from the hypothalamus. And it continues this way until puberty, at which point the hypothalamus becomes less sensitive to the inhibition, causing gonadotropin-releasing hormone levels to increase. GnRH stimulates the HPG axis, and the blood testosterone level will begin to rise, triggering spermatogenesis and the appearance of those secondary sex characteristics. In most boys, this is between 12 and 14 years old, but it varies. Usually within a few years after the onset of puberty, hormone levels stabilize, and as soon as spermatogenesis begins, males are capable of sexual reproduction, even if those hormone levels are still fluctuating. Now, I keep saying secondary sex characteristics, but what are those? Well, they develop primarily at puberty and may continue to develop into early adulthood. But these include growth of pubic, axillary, and facial hair. So that's groin, face, and armpit hair. There's enhanced hair growth on the chest or other body areas in some men, an enlarged larynx and thicker vocal cords, a noticeable laryngeal prominence, and a deepening voice. So the Adam's apple and a deep male voice tend to result here. Thickened skin and increased secretions of the sebaceous glands. This oilier skin tends to result in more acne for men. And testosterone can cause some somatic effects as well, most of which are anabolic. Now, Under the influence of testosterone, you're going to see more calcium salts deposited in bone, making it denser. In addition, skeletal muscle mass will increase, accounting for higher muscle mass in males compared to females, and all organs in general tend to be larger. And this accounts for the male's larger cardiac output and total lung capacity. And testosterone also has a direct effect on erythropoietin production, causing males to have a higher number of red blood cells than females. Finally, testosterone does tend to have an influence on behavior. For example, it's the basis for the male libido, which is the desire for sexual activity. However, the long-held idea that testosterone is responsible for aggression is really highly disputed, and not much evidence really suggests that it is. In fact, that tends to just be um, more directly related to underlying psychological issues, and testosterone isn't the cause, but it is rather an exaggerator. Now, there is male climacteric uh, uh, a period of time. This is called andropause, and this is in which reproductive functions begin to decline, and these changes are generally gradual and begin early in their early 50s. May or may not be noticeable changes at all in most men. Um, the testes change size and weight. They may decrease, and the number of sustentacular interstitial cells begins to decline. With sustentacular cells producing less inhibin and interstitial cells producing less testosterone, gonadotropin releasing hormone increases the production of follicle stimulating hormone and luteinizing hormone, and so that negative feedback loop decreases. But if FSH and LH levels remain high, it indicates that the testes are not producing enough testosterone to suppress their function. I'm sorry, to suppress their production. And so an increased number, a reduced level of testosterone results in their gradual production of fewer sperm cells. Although this is not absolute, many men in their 70s or 80s have fathered children, and in addition, a low level of testosterone has is associated with a depressed mood and fatigue in some men. And this can lead to a decrease in muscle strength and decreases in bone density. But bone density tends to decline more in age anyway. In men, bone density typically declines more slowly in men than in women, and this is because the loss of testosterone is more gradual. But the most noticeable change for men during climacteric is the enlargement of the prostate, and this compresses on the prostatic urethra and can cause disurination, difficulty with urination. And that is the old adage: men will sometimes have trouble urinating, or they will frequently urinate as they become elderly. Now we're going to round this out with some more test yourself questions. What is the maturation process in which spermatids elongate and shed excess cytoplasm? Maturation process in which spermatids elongate and shed cytoplasm. That is spermiogenesis, not to be confused with spermatogenesis. Now, you see a male bodybuilder taking testosterone supplements, increases his muscle mass. But after 3 weeks, which of the following do you expect to observe? So this guy has excess testosterone in his body, right? Excess testosterone is going to negatively impact that feedback loop, so there's not going to be as much gonadotropin releasing hormone produced. 75-year-old male has noticed he's had difficulty urinating. His physician says his difficulty is most likely due to what enlarged gland? He's an elderly man. He's probably having difficulty with his prostate being enlarged. Remember, when the prostate swells, it pinches the urethra. All right, that's all we have for this one. I will see you guys in the second half. We will begin talking about the female reproductive system.